Alright, everybody. Today I'm going to talk a little bit about the sort of two, what I call chemical senses, taste and smell. And in general, what I'm hoping you take away from this as main points are, we can understand, we talked a lot about what an action potential is and how that transmits a signal down an axon, but it may not really be clear to you yet how an action potential is transmitting information. I think that's easier to see in a sensory system, and these two different systems do so in two different ways. You should understand what is meant by a chemical sense and how these will end up being different from our other senses. And in general, for all the senses we talk about, you should understand what is being sensed in particular, like physically, how is that detecting information from the world, and where's that going to in the world. Right, so how many people have seen this sort of taste map before? This one is not labeled, but typically we talk about sweet being at the tip of the tongue, bitter at the back, and I think sour and salty mixed in there at 2 and 3. I always start off here first saying this, if you learn about this, is wrong, right? So our tongue in taste does taste these different components of taste, but this idea of a taste map where there's certain parts of your tongue that respond to certain taste is not true. All parts of the tongue respond to all parts of taste, right? And it does so in these little mini organs called taste buds, right? If we look at this, it's sort of a cross section of the tongue, right? And within our tongue, we have these little pores, right? And these are your taste buds, and below them is sort of where the real stuff happens, right? So when you're eating food, you're breaking it down, little bits and chemicals that come off that food go into these taste buds, and then inside these taste buds are the cells that actually detect it, right? And these sort of fluorescent images can show that here. It's sort of packed with these different cells, and right at the tip here is where they sort of really concentrate the receptors on those cells that are able to detect different aspects of taste. This is a sort of over the top for this course, but just know there are different types of neurons in there that are taste receptors, and that they can vary based on what aspect of taste there is. Some of them get reused, for example, for sweet, umami, bitter. It's all the same receptor, the same cell, but with different receptors on it, whereas salty and sour seem to be their own cells. Right. And within one taste bud, you can have the receptors for each one of these. These are not specific to one taste bud. I always like to point this out because it nicely sort of shows where these taste buds are, but also brings up sort of a neat little factoid, I guess. So most of us are what you consider non-tasters here at the bottom, and this shows our tongue, where if you can sort of use these different chemicals or dyes that are non-toxic, and it can show you the little pink spots that are left. Those are your taste buds, right? So kind of all over your tongue, fairly sparsely here compared to this person here who's called a super taster. They tend to have almost 10 times as many taste buds, and they literally are able to taste sort of more intensely and more with higher discernibility. These people can actually, this can be like an employment thing, right? I've heard I know the story of one guy in particular because he donated a lot to the lab I worked in grad school. He basically got his first job at Pillsbury because he was a super taster, and he was a taste tester for Pillsbury. That was like his job. He actually worked his way up to CEO, basically. Right. So, how are signals from these taste buds transmitted to the brain? So this brings up the first way this can be done, and it's sort of the simplest way to transfer information, and probably what we think of when we think of that. It's called labeled line transmission. So this is so first off, taste is a chemical sense. So like I said, there's all these different chemicals, and we haven't really talked about this, right? This is sort of the five main things in taste we can sense. We'll talk about in a second, but maybe it's up to 2 more. But bitter, sweet, sour, salty, which is all just salt, and then one that we call umami, which you may not have heard of. That's basically sort of protein, right? Savory, things that are rich in protein. So it's called a chemical sense because it senses a chemical, right? And these physical molecules have to bind to these receptors, and we sense that, and that's what we're getting when we taste something, right? And this will be the same thing for smell, that there's actual volatile chemicals in the air that go into our nose and they touch physically the receptors. So it's these chemicals, right? As opposed to something like touch or hearing, which are actually mechanical senses. There's a physical movement that's causing those, or vision, which is kind of the same thing, where it's detecting light or photons. That's a little bit different in itself, right? So when we think about all these things, they all use a similar strategy, but they're all able a bit different, and you should understand that across their senses. So for taste, we would use what's called label line, where right within this taste bud, we have different receptors for all 5 of these different ones, and then they're going to end up connecting the cells and putting out axons that go directly to specific parts of the brain. And the brain will know, oh, this axon, it's been labeled bitter, this axon umami, and so forth. And that's why it's called label line, because it knows that if it's firing, if this axon firing, that means you're detecting bitter. And if this one's firing, it's sour. It's a fairly straightforward way at which we can get these sensations to the brain. Now, when it goes to the brain, right, it's going from the tongue, and the key thing here is that every sense has a primary sensory organ. Remember when we talked about the somatosensory cortex, right? That'll be the primary sensory cortex for touch, right? That's the first, the place that the signal goes straight to. And it's usually, we think of these cortexes where the pure signal is, right? It hasn't been processed, it's getting the straight information here. Now, that being said, in the case of taste and some of the other ones, there are some sort of relay steps. So you can see this where taste is going from the tongue to the geniculate ganglion and a number of other ones, but the primary cortex here is what we call the gustatory cortex. Gustatory is sort of another way to talk about taste. Gustation, you may have heard of, for digestion-related things, right? So this is the main area of the brain where that goes. In addition to the gustatory cortex, the other main region you might hear about is the solitary nucleus, right here in the NST, as being the sort of primary relay to get to here. Now, this has an important, for a number of different reasons, but in particular, lesions here can be lead to difficulty in swallowing, and there's a feedback loop where, quite often the thought is that if you have a taste that makes you lead to vomiting or makes you want to vomit, that's because of activation in here, where it makes sense that we would tightly control taste and sort of our ability to swallow, or sort of the opposite of swallow, right? And so if we look at a human brain, and we'll look at a sort of more detailed one here. So right here in sort of this, the insula here, right, where this part of the brain folds over, is where the main taste reception here. And they call this the taste area in this one. I think we can see it more clearly in this next image where it has a sort of a cutaway, right? I always think of these, as sort of the arms that come around the brain, right? And you kind of have to look in. So this actually is a sort of deep fold into the brain called the insula, and the gustatory cortex is often referred to as the insular cortex, right? That is a synonym for them, right? So right in here is where the real gustatory cortex is. And inside of that is where the taste map is, right? And this will be a sort of common feature across pretty much all of our senses, the idea of how these different components of a sense are sort of mapped out, right? So there's a really nice paper I always like that shows basically they stuck a very tiny little microscope into this insular cortex, and they could do this in a living mouse, and then it had one of these calcium sensors in, so they could look at the fluorescence, and they would give that mouse different foods, bitter, salty, sweet, and so on, and then they could sort of look and see, are there neurons in certain regions that respond? So in this region, they found the neurons are highly sensitive for bitter, much less so for anything else, right? These neurons, there's a scattering of them. And they can see a high percentage respond to bitter, very low percentage for the other ones. They were able to create this taste map in the brain where those labeled lines that are coming in, right, seem to be ending predominantly in this region, right? And like I said, there's regions for each of the other senses as well. Right, 5 main tastes: sweet, salty, bitter, sour, umami. 2 potential others, and we're still a little on the fence on these to some degree, but CO2 seems quite likely that we can taste that. You maybe have a sense of that with soda, right? It tastes different when it's carbonated because we're probably detecting that CO2. Also, fat probably has its own, but that one's a little more. Right, this is a question we can come back to later in class, right? So, here is a little more deep dive on these. And so, umami, sweet, and bitter all use a similar set of receptors in that they use what, remember earlier, these are G protein coupled receptors. Umami and sweet actually use half of them. It's the same. You see it says T1R3. It means half of this receptor complex is the same receptor, and then the other half is either R1 or R2. That makes it either umami or sweet, right? Bitter, sort of interesting. Most of these have one receptor for one taste, right? The sweet receptor basically binds to sugars. That's sweet, right? Umami basically binds to amino acids. That's, protein, meat, savory. Bitter is actually a sense of many different molecules, and this seems to be sort of bitter is traditionally something we don't like, especially very strongly. It seems to be detecting molecules that are not healthy for us, right? Or that are maybe toxic or poisonous. And that's actually a group of 30 plus different variants of a similar gene that detect a whole range of different chemical compounds and things that all give us this bitter sense. The most pure bitter you can really think of is something like quinine, which is just found in tonic water, or another one that I would, in a different year, give people to test in class is if you get unsweetened chocolate, that's very bitter. Sodium is actually, it says here, the ENaC channels that seem to do, we actually, these ones that are in the red box are less certain. We're not 100% sure on what the sodium channels. This is one of the better guesses here. And then here are channels for sour and carbonation, but I do want to mix up. This actually is probably not the correct one. And in recent years, this different channel called OTOP1 seems to be the clear favorite for what is the sour receptor. But I'd like to point this out that this is probably something I would have thought if I was in your shoes. Like, we probably nailed this down like several decades ago, but we're still trying to figure out what these chemical receptors are for the different senses. I want to mention this because I think this is an interesting thing, and sometimes we get to do this in class, obviously not this year, but there are these berries called miracle berries that don't have any taste themselves, but we actually use them to sort of modify your taste. So basically what they do is they make sour things taste sweet, and you can use them, for example, to make, in general, people think they make almost everything taste bitter, right? So then we can use them for things like cancer patients who need to eat but don't have much appetite. This will make the food more new and interesting and increase how much they eat, right? Often this is referred to as taste stripping, right? You can order these tablets of these off Amazon and try it out for yourself too. Now, here's a neat experiment I think that shows sort of proved how this actually works. So there's a chemical within this called miraculin, and what they did is they had cells that aren't actually neuron specific. These are just sort of generic lab used cells. And what they did is they put the channels for that for sweet in here, right? They're for sweet in here. And what they did then is they measured basically how active they were at different levels of acidity. So acidity is basically sour, right? If something is low pH, it's more sour. And what they found is that in the presence of miraculin, these sweet cells, right, they should only really be responding to sugar, now are also responding to pH, which that sort of makes sense. If you think about how label line transmission works, if you trick a sweet cell into being active in sour, The brain's not going to know that's sour, right? The signal that is labeled to that sweet cell is sweet. So if you activate it with the wrong signal, you get the wrong activation, but your brain's going to think it's sweet, right? And this is just sort of the schematic. What is less clear about this, though, is that if people who taste this, you'll also notice that the things taste less sour, and it's unclear how that works. All right, so let's move on to smell. Smell also a chemical sense. Right, if we look at the cross-section here, right, this is your nose here. We all know that's important for smell. I find this sort of interesting. So right here, right, this is the brain, and if you see here, notice before, right at the bottom here, there's this sort of little line of cells called the olfactory bulb, right? And it puts little projections through a part of your skull into your nose that have little perforations in it, right? It's called the cribriform plate, and it sends these projections, you can compare like into your nasal cavity, right? And this is where the detection of smell comes from, right? All these little sensors are here, and as you breathe in, comes the chemicals from the air interact with here and send signals into your olfactory bulb, right? The receptors for this work sort of similar, right? It's a chemical sense, so they're binding chemicals, but it's sort of also very different. And the person who really, I think, gets the credit for figuring it out is Linda Buck, who won the Nobel Prize for this in 2004. Where basically, as opposed to taste, where we have just a handful of receptors, in humans, we have approximately 600 of what we would call sort of classical organ receptors. There's ideas that we might have another sort of additional 400 sort of weird ones that we don't understand well, but in mice, they actually have 1,000 or more of these different receptors, which I think indicates strongly that to mice, odor is a much more important sense. So the idea here is that these now we can pick up a much wider range of smell. We can smell many different molecules compared to how many we can taste. This then becomes even more information through something called combinatorial coding, which I think is better talked about here. Right? So if we simplify this down to, let's say, 14 receptors shown on this diagram, we can have scents that just activate one receptor. I love the description here because I'm never really sure what this means. But this molecule here, they're saying only activates this odor receptor 5, and that gives you rancid, sour, goat-like. I never know what that means. But, right, you can see, for example, this 1D. It still activates receptor 5, but now it also activates receptor 6 and 2. So that now gives you violet, sweet, woody, distinctly not goat-like, I guess, right? And you can have a whole variety of these. So now, label line is not important, right? Like, connecting this one receptor to the brain is not important for lots of things. You need to figure out how to combine all these, right? So that provides a ton more information than just, the 4, 5, 6, or 7 different tastes we can get. Now we can detect, literally potentially more than 1 million, right? Because it's one of these things, if you do the math, if we have 600, how many combinations you can have is really like 600 exclamation mark. And I don't know what that leads up to, but there's good bets that for sure, like realistically, we can detect at least hundreds of thousands, if not over 1 million different scents by using this combinatorial or pattern coding, as it may be, right? Which I think you can see some similarities, but it is a distinction compared to a label line. It's much. The label line is very simple, distinct. You know what sour is, and sour is not, almost something else, right? Whereas these, you can see that, the lines can blur a little bit. For example, if you look at some of these that are very close, like H and E, it's really just varied by 1. You can see how those lines will get blurry a little bit, but this also gives us a much greater repertoire of smells we can get. Right. Think about that question. I think that will be important. So, what regions are important for olfaction? So if we're looking at the brain bottom up here, right, this is the brain specifically at you. Here's the olfactory bulb that will be dropping those sensory neurons into your nose, and it goes back to there. So the olfactory bulb is very near and leads directly into the primary olfactory cortex, which you might expect, right? That's the initial one here. And it's a fairly simple circuit compared to all of these. This whole thing is called the olfactory tract. A couple other areas to mention here is, right, and we talked about this here, amygdala, we looked at here, is very close to this olfactory bulb. And I think that strongly ties in why scent is very closely tied to memory, and also why fear, in particular, and smells go strongly together. Our brain seems to be hardwired for things like fire, smoke smells to cause fear for probably good reason. Right, and that's right. A couple odds and ends to talk about in regards to scent. Pheromones, if you haven't heard of these before, it's really these sort of other scents that are used in sort of detecting emotions or who somebody else is. Now, these are very important in animals. They're present, the whole organ system seems to be present in humans. They're actually, I have the toughest time with this name. Volar nasal organ, right, is the detection organ. The up here is the olfactory one, is much closer to the nostril. There's a small organ. We seem to have this organ. It's uncertain how functional it is, right? In other animals, it's key for things like mating, predator detection of friend versus foe. So, you know, detecting if another animal wants to mate with you can literally do this by smell. And if the predators nearby, that would activate this, and it's a much more base instinct level of sort of smell that can directly detect sort of fear and things like that. And then very interestingly, like you can this is very obvious in mice, where a few mice that are brothers, male mice in particular, are very territorial, and brothers are okay with each other, but if you put for example, mice in a lab that were born separately and put them together, they're going to fight almost instantly. It's because they detect that they are not sort of friends based off of that. Right, so two brain regions that are linked, and I think this should be obvious. Hypothalamus, right? That's a lot of these. Remember the 4 F's there, right? And amygdala, the fear, right? An emotion, I guess, possibly in general. It's very closely linked to the limbic system. Here's a question to think about that we can talk about. Right? So hopefully this is something that we keep on looking at. Right? Holding your nose, if you have a cold like I do right now, clearly affects your ability to taste things. Everything gets more bland. And I think you can see that our sense of taste is pretty limited. So our sort of total perception of how food works is based on both of these senses, and probably much more so by smell, right? We can have a much larger repertoire of chemicals we can detect through smell. But something like salt, I always like to mention, what does salt smell like? Right? It doesn't. And so the key distinction here is that smell works great for volatile chemicals, basically chemicals that diffuse in the air, right? Gasoline, right? That's something that has very strong car exhaust, right? Strong smells. Fire, things like that. But something like salt doesn't. So chemicals that are much more dissolve or move more freely in liquid or water, right? Like salt or sour, right? Like what does sour smell like? That's a much stronger related to taste. All right. So last thing I'm going to talk about, and this one will definitely come back in class because it's always interesting, right? I think everybody probably has their answer, has the dog or a human have a better sense of smell, right? I'm going to guess we're all thinking dog right now. That's what I would do. But I'm going to show you this video and talk you through it that I think is pretty interesting. Hopefully, it sounds good.
The scent tracking arena is shown from above. The arena is 10. The scent tracking arena is shown from above. The arena is 10 meters on a side with a 10-meter.
So I'm going to let them talk.
For this demonstration, the trail is made of highly visible rope. During the actual experiment, the trail is not easily visible. The trail is composed of 2 straight segments connected by a 45-degree angle. The subject is outfitted with goggles, earmuffs, gloves, and knee pads to block non-olfactory input. The subject wears a data logging device in a backpack to record their sniffing. The nasal cannula used to measure sniffing is seen here. Here the subject is wearing nasal prisms to alter the spatial reach of their sniff. At the start of a trial, the subject is positioned facing the odor trail and has been instructed which direction to turn on encountering the trail. Beyond this initial instruction, the subject is given no further information about the path of the scent trail. An example tracking run is shown here at roughly 4x speed.
All right, so if you didn't quite get it right, the whole idea is these experiments, right? They block out all their other senses, and this one, they used undergrads at Stanford for this experiment. And what they did is on this trail, they didn't have this line here, this is really for demonstration, but what they did is basically took a chocolate bar and wiped the trail. They could have went this way or could have went this way. And they just had them using their sense of smell because everything else was blocked. And they might have modified the sense of smell in different experiments using those cannulas that they talked about to sort of expand it. But they took, either undergrads or dogs and saw who could do this experiment better. And the interesting thing is about 2/3 of dogs and 2/3 of humans did this accurately, which I think the take-home point is that basically they, we both have the ability to smell. It's really much more how do we interact with our environment that's different, right? Smell is not our primary sense. Vision very much is for us, but for many other animals, smell is, and that's why, a dog's and a mouse and a rat's nose is much closer to the ground at times than at almost all times than ours, and that's, probably the bigger difference. It's not something physically biological about it. All right, so that's what I have for you on taste and smell. We will talk about it a little bit more later.